So, have you ever really thought about calcium? You probably just think of it as the basic building block of your skeleton, right? Just sitting there, making your bones hard. But the truth is, calcium is this incredibly active, highly volatile ion that acts as the literal electrical wiring for your entire body. Without a strictly regulated supply of calcium flowing in your bloodstream, your muscles wouldn't contract, and your nervous system would just completely misfire. The margin for error here is essentially zero. So what maintains this high-stakes biological tightrope? Well, it all comes down to a master biological switch.
And that critical master switch? It's the parathyroid gland. In this explainer, we're going to do a first principles breakdown of exactly how this elegant endocrine system operates. We'll explore the cellular mechanics of how it senses minute changes in your blood, how it literally alters renal physics to maintain balance, and what happens to the human body when this whole system tragically breaks down. Here's a quick look at our roadmap for today. 1. The calcium balancing act. 2. Inside the parathyroid gland. 3. Receptor mechanisms. 4. Biological actions. 5. Primary versus secondary hyperparathyroidism. And finally, 6. Clinical signs of system failure. All right, let's get into it. Section 1. The calcium balancing act. The parathyroid glands, which sit just posterior to the thyroid gland, are the absolute primary regulators of our calcium physiology. They basically have one main job: producing parathyroid hormone, or PTH. When released, PTH acts like this highly targeted biological missile. It coordinates directly with your bones, your kidneys, and your gastrointestinal tract to make sure blood calcium remains incredibly stable. Because major disorders here lead to really dangerous extremes. You can get hypercalcemia, or on the flip side, hypocalcemia, which is a drop in calcium that causes severe electrical instability in your muscles, resulting in these dangerous involuntary spasms known as tetany.
Section 2. Inside the parathyroid gland. Anatomically speaking, we typically have 4 and sometimes even more of these tiny parathyroid glands tucked right onto the posterior surface of the thyroid gland. If we zoom in microscopically, the cellular factories driving this entire operation are made up of 2 important cell types: oxyphil cells and parathyroid chief cells. Actually, it's the chief cells that are constantly monitoring the blood and synthesizing our key player, parathyroid hormone.
But how exactly is that hormone made? Well, PTH doesn't just pop out fully formed. It's synthesized as a much larger precursor molecule called a pre-pro-hormone. Clinically, this hormone circulates in 3 distinct forms. You've got intact PTH, which is the full, active molecule, alongside carboxy-terminal and amino-terminal fragments. These specific pieces basically dictate how the body communicates with and ultimately clears the hormone from your bloodstream.
Moving on to Section 3, receptor mechanics. So, how does the parathyroid gland actually know when your calcium levels drop? Enter the calcium-sensing receptor, or CaSR for short. This is a G-protein-coupled receptor where ionized extracellular calcium acts as the primary ligand. You can think of it kind of like an incredibly sensitive thermostat that uses strict if-then logic. If the CaSR detects a drop in extracellular ionized calcium, then it immediately signals the chief cells to release parathyroid hormone from their vesicles. And this exocytosis, it happens blazingly fast, in mere seconds to minutes.
But calcium isn't the only regulator here. Extracellular phosphate plays a massive role. High levels of it actually affect PTH messenger RNA stability and promotes the growth of parathyroid cells over the course of hours and days. Meanwhile, calcitriol, which is active vitamin D, does the exact opposite. It exerts an anti-proliferative effect and literally inhibits PTH gene transcription. And finally, you have FGF23, which steps in to drive the loss of phosphate right into the urine.
Once it's released, PTH has to actually bind to its target organs, right? That happens via the PTH1R receptor. This is a highly complex G-protein-coupled receptor that is heavily expressed in both the kidneys and the bone. The binding of either PTH or PTH-related protein kicks off multiple intracellular signaling cascades. It basically acts as the mechanical lever that physically changes the cellular behavior.
Now, while PTH1R does the heavy lifting for calcium homeostasis in the bones and kidneys, it's pretty fascinating that the body utilizes a whole secondary network for this hormone too. The PTH2R receptor specifically binds PTH and is found in high concentrations in places like the central nervous system, the gastrointestinal tract, the lungs, and the testes. On top of that, there are non-classical receptors that interact specifically with those little PTH fragments we mentioned earlier.
Let's look at what all this actually does in Section 4, Biological Actions and Effects.
When PTH successfully binds to its targets, it initiates 4 really powerful kinetic actions to drive blood calcium back up. Step 1, it increases bone resorption. It literally signals the bones to break down their mineralized matrix and dump stored calcium directly into the blood. Step 2, it completely alters renal tubular physics, dramatically reducing urinary calcium loss, so your kidneys hold on to whatever calcium is already floating around in the system. Step 3, it forces phosphate loss in the urine, which prevents that phosphate from just binding up the newly freed calcium. And Step 4, it increases intestinal calcium absorption, a process heavily mediated by active vitamin D.
This entire system relies on a wonderfully efficient use of biological hardware. The classic endocrine loop uses the PTHR1 receptor in the kidneys and bone to drive up blood calcium. But get this, the exact same receptor is utilized locally by a completely different molecule, PTH-related peptide, to regulate growth plate cartilage in developing bones, smooth muscle, and even breast tissue. It's serious multitasking.
All right, Section 5, primary versus secondary hyperparathyroidism.
So, hyperparathyroidism is a generalized disorder of this whole metabolism, and it's divided into distinct categories. Primary hyperparathyroidism is usually an internal hardware problem. The parathyroid gland itself just goes rogue. 80 to 85% of the time, this is caused by a benign solitary adenoma that's just pumping out unregulated PTH. Conversely, in secondary hyperparathyroidism, the gland is actually behaving normally, but it's being forced into overdrive by an external systemic trigger. Most commonly, that's chronic kidney disease, profound vitamin D deficiency, or severe malabsorption.
Looking at the epidemiology, this primary, unregulated overproduction of PTH is actually much more common in women, and it typically peaks in the 5th to 6th decades of life. What's super interesting here is the underlying genetic architecture. These rogue adenomas are often linked to specific chromosomal defects. Basically, the cellular blueprint itself in genes like MEN1 or CDC73 has been fundamentally altered, which causes the glands to completely ignore their normal biological breaks.
And finally, Section 6, clinical signs of system failure. When you've got excess parathyroid hormone flooding the system, it causes a massive clinical cascade. Because PTH is aggressively ripping calcium out of the skeleton, you end up with severe bone disease. And because it forces the kidneys to process huge, abnormal amounts of minerals, you get nephrolithiasis, which is just a fancy word for kidney stones. All this downstream metabolic stress can also lead to proximal renal tubular acidosis, gout, and even anemia. Yet, despite that terrifying list of symptoms, one of the most common clinical manifestations today is that the patient is completely asymptomatic. It's often just caught by routine blood work showing high calcium. But when it is symptomatic, medical professionals use a classic, rather grim mnemonic to remember it: bones, stones, and abdominal. We also have to remember that this condition can be tied to familial conditions, and crucially, it can be pharmacologically triggered. Both thiazide diuretics and lithium therapy are known to alter this incredibly delicate calcium balance and force the parathyroids into a hyperactive state.
So, we've tracked the incredible journey of calcium regulation today, all the way from a tiny G-protein-coupled receptor on a single parathyroid chief cell, out to massive systemic changes affecting basically every bone and organ in the body. It leaves us with a pretty profound realization. If a microscopic receptor glitch can literally rewrite our bone density and kidney function, what other massive biological levers are hiding in plain sight within us, just waiting to be understood?